This video will cover what we know about cells from the scientific area of cell biology. Cells are marvelously complex building blocks of life and are presented at this particular point just ahead of the video on proof for evolution so that you will have a greater understanding of why cells, their existence, is a negative for evolution theory. Darwin was very much aware that if complex things were found, that they would negate evolution theory because cells could not evolve slowly over time. They're so complex that it's impossible to think that they could ever evolve slowly over time. Now, here are some of the statistics that we know about cells. Number one, the human body contains about 100 trillion cells. The estimates vary between 50 and 100 trillion, but somewhere around that number, trillions of cells contained in the human body. There are a number of different types of cells. Over 200 is the general number that people talk about, but somewhere between 150 and 200 in the literature. And there are different types of cells. We have stomach cells, kidney cells, skin cells, bone cells, liver cells, all sorts of different types of cells, and they perform different tasks in the human body. Now, cells don't live forever. They die and are replaced at different times, different ages, depending upon when they need to be replaced due to the difficult life that the cell has gone through. In particular, some of the examples here are that stomach cells are replaced every 2 or 3 days. We give our stomachs a rather difficult time with the various things that we eat and consume inside of our intestines, and the cells in the stomach, therefore, need to be replaced every couple of days. On the other hand, bone cells are replaced every maybe 25 years or so, and so there's quite a range of time span and during which cells are replaced, and all the various other types of cells, kidney cells, skin cells, are replaced at intervals in between a few days and many years. Now, one of the most amazing things, or the most amazing thing to me about cells, is that each cell contains a complete set of the body's DNA blueprint. In other words, all of your about 100 trillion cells contains a quantity of your DNA, or all of your DNA, that enables the construction and replication of every cell in your body. In other words, each type of cell does not just contain a blueprint to make a new copy of itself. It contains enough information to make every cell in the body, and that's just amazing. It's kind of, in some sense, overkill, but it is a necessary way that the cells have been structured such that each cell contains a complete set of your entire DNA. Now, a cell itself, we'll see in the next couple of slides, is a complex thing, and each cell contains the nucleus of the cell, we'll see in a minute, contains about 50 workrooms. There are things like enzyme manufacturing plants, quality control plants, enzyme folding plants, enzyme folding quality control plants, and a whole bunch of other types of plants that are sub areas within the nucleus of a cell to do the functions, perform the functions required of that individual type of cell. And it is this complexity that is the reason why cells are negative for evolution theory. Now, here's a look at the basic cell structure. We have an outline here of the outer wall of the cell, and that defines the cell, its outer wall. And then there is an inner area here that's called the cell nucleus, in which the bulk of the business of the cell is conducted. So there's an area in between the outer wall and the nucleus that performs a very important function, or really the initial function. The bloodstream supplies food to the cell, and it enters in various places through the cell wall, and that food is in form that needs to be converted into energy that can ultimately be used within the nucleus of the cell to perform its tasks. The conversion plants are called mitochondria, energy conversion plants, or mitochondria for short, and they perform this important function of altering the food that comes in to just the right form needed by the cell nucleus. When we look at the DNA video later on, it will be very important to consider mitochondria or mitochondrial DNA. This is a small quantity of DNA in these mitochondria plants, and it's very, very important, as we'll see later on, in determining the first human ancestor, but that's a later subject. So if you think of this cell as being a manufacturing plant, in effect, this area between the outer wall and the nucleus is kind of the shipping and receiving area. Food comes in, is converted, it enters in here, and at the same time waste comes out and is returned to the bloodstream where it is removed from the body. So that's the general structure of the cell, and we'll look at the next slide at the behavior, the complex behavior of this region called the nucleus. This slide shows in some detail the behavior of cells as they operate to do some particular function. It's a rather busy slide, both the picture and the words, and at the end of this discussion I'll give you a chance to stop the slide to take a look at it. Let me go through a brief description of what you're looking at here. So when a cell is requested to do something, an order arrives from the brain. So the brain sends a message, I've shown it here as a letter, sends a message to a particular cell or a group of cells that you need to do some function, you need to perform something that I want done. That function is performed by an enzyme. Now enzymes are nothing but organic molecules that are the main working molecules within cells. There are a couple of other types of working molecules, but the main one is an enzyme that performs the function that the brain has requested. So the first thing that needs to be done is that the enzyme needs to be manufactured. So there's a plant here. These various plants in various locations within the inner nucleus here are called organelles formally, but let's just call them plants here, the things that perform functions. The first thing that that plant needs in order to manufacture that enzyme, it has to get the plans for it. So it goes to a particular chromosome in the DNA, a particular part of the DNA, gets the plants. A copy of this is sent over through something called messenger RNA. So the plans are sent back to the plant in order to manufacture that enzyme, and it goes about manufacturing that enzyme using the energy and the molecules and the atoms that are within the nucleus to assemble that particular enzyme. So that enzyme is constructed. The next thing that happened is that a molecular transporter, I've shown it here as a truck, takes that enzyme and transports it to another plant, and that plant is the quality control plant for that enzyme. So that quality control plant, of course, had to have the plans as well, and it checks to see whether the correct enzyme has been made. If it's not been made correctly, it's recycled, but if it's made correctly, then it passes on to the next stage and ultimately would do that. So another truck, another molecular transporter, transports that enzyme to another plant, and that plant is called an enzyme folder plant, or a plant that takes that enzyme, that long molecular polymer, and folds it in just the right way. Now, why does an enzyme need to be folded in just the right way? Well, that particular enzyme, say if it just happened to be requested by this plant here that needs the enzyme to perform the requested function from the brain, it has holes in its wall, or doors in effect, and the doors are in just the right shape so that just the right folded enzyme can enter into that door. And so this is very important that this enzyme be folded in just the right way to get into the intended plant. There are going to be potentially a lot of enzymes within the nucleus at any given time, and we don't want the wrong enzyme getting into the plant that needs the right enzyme. So the quality control, the folding of the enzyme is very important, and then that folding process says as a final plant here, the quality control folding plant, to see whether that enzyme has been folded correctly so that it can enter into the correct location. Well, once the enzyme passes all of these tests and successfully gets through the quality control folding plant, it is then finally transported by yet another molecular transporter and enters into the plant wall just where it is needed, and the cell then at that point can perform the function that it was requested to perform. So that's a relatively simple description of a complicated process that goes on, and understand that there can be multiple enzymes generated at basically the same time doing various things within the cell, so it is indeed quite a complicated process. At this point, let's just back off a little bit and think about if you had a cell or cells in your body that were sick and you needed some pharmaceutical medicine, some chemical to come into your body to cure whatever was wrong with those cells. Well, what would the pharmaceutical research company have to do? Well, they'd have to, first of all, get something in the bloodstream, have that molecule enter into the location of the in the cell wall, have it able to get into the cell nucleus, and have it able to maneuver around. And let's just say if this was the same plant that needed that pharmaceutical medicine, that they have it folded in just the right way that it can enter into that plant and do what the intended task is. Now, it's as I've explained, the normal functioning of a cell, where all of the folding and moving and all that is part of the original design of that cell. It's one thing to have all that work as designed, but it's another thing to come in kind of on top of that and design another enzyme, another molecule that's going to get in there and kind of work in between all these various other things that are going on. I think from just that brief explanation, you can understand why it can take hundreds of millions of dollars and many, many years of research to develop a medicine that will do just the right thing and enter in just the right location. You have to first understand what cells need it, what all the parameters are in terms of the cell door sizes and all that, and then you need to investigate a wide range of molecules, of chemicals that can be converted into a pharmaceutical medicine that will be able to do the intended function. It's not too hard to understand why that is not a perfect process. And in fact, if you have watched commercials on television and have seen, the wondrous things that they do, their intended thing, and then at the tail end there's this long list of side effects that they just rapidly say can cause this and that and this and that and that. Well, one of the explanations for these side effects is that these pharmaceutical medicines get into this cell and perhaps go to the right location, but some of them go to other locations and get into other plants, and indeed some of them go to other cells that are not in need of that pharmaceutical and cause these so-called side effects. So that's just kind of an interesting sidelight to add to this explanation of how cells work, so that you can appreciate indeed how extremely complex they are, and how difficulties can result as a result of medicines that are trying to get into the middle of this complex process. At this point, we'll move on to the last slide, and I'll just give you an instant here if you want to stop it and look a little more closely at what we've got here. Let's look briefly at what we can conclude as a result of looking at how cells behave and how they perform the functions that they are intended to do. Cells are, first of all, wondrously complex. I think I've shown you just a very simplified version, but yet it was very complex as to how cells behave. And yet they're extremely efficient over the bulk of our lifetimes. We'll see in a later video on telomeres and telomerase that cells ultimately break down, and ultimately toward the end of our lives are not functioning as well as they did during the bulk of our lives, and it's unbelievable that they can perform as well as they do for as long as they do, in my view. Now, it's obviously the reason why I presented this video on cells ahead of the next video, which is proof for evolution, is so you can get a more complete view of what cells are like before you see the negative result for evolution. The basic idea is that cells this complicated, this ingenious in their behavior, could not have evolved very slowly over time as required by evolution theory. It is impossible to have happened. And indeed, if you saw the scientific method video, I did mention there that Professor Anthony Flew, a 60-year number one atheist in the world, became aware of the information that I just showed you about how cells perform and do what they do. And it was the thing that resulted in his last book titled There Is a God, in which he realized that the cell could not have evolved slowly over time and had to have been had some design going into its construction. In any event, let's move on now and look at the next video, which is proof for evolution.